have Dr. Hagen Tilner. Sorry, I just wanted to make sure I wasn't muted. We have Dr. Hagen Tilner, who's an assistant professor at the BMRI, the Brain and Mind Research Institute at Weill Cornell. His lab, which is really at the intersection of molecular biology and computational approaches, which through these things like these hackathons, we're really trying to promote. They study single cell isoform expression in the brain. And I'm not going to try to explain what that is because I have no idea what it actually means, but I'm really excited to learn more about this talk. And sort of as Bobby's mentioned, if you are interested in this type of work, Dr. Tilner is looking for a postdoc, so be sure to reach out to him about that. Thank you.
Cool. So let me share screen up and go into full view. Right, can you see that? Is it now visible? Okay, great. So I'll put the videos away. So yeah, thank you. Oh, damn, this is not working. Thank you for the kind introduction. And so I like to start my talks by showing this this cover that we made in 2012. And when I say we, it was actually Luisa, who was my wife. She wasn't at the time, but now she is. And where we try to represent a lot of papers that were published in the framework of the ENCODE project. And so I'm not going to go into all of it because there were multiple papers, but I'm just going to mention what's really important to the paper that I was involved in and that we try to represent. That was sort of the starting point of this cover, and then everything else was added on. So here you have the DNA template, RNA polymerase making RNA, the messenger molecule. And these rocket-shaped things here are exons. And what you see is that between these two exons, the intron is already gone. So it's basically no intervening intron. Between these two, it's gone as well. Here, it's gone as well. Then you see two exons that are still separated by an intron. Then again, it's gone here, and then another one is there. But the statement was basically that the majority of introns are removed from these RNA molecules while the polymerase is still producing the downstream parts of that RNA molecule. So in the splicing jargon, we would say splicing happens co-transcriptionally, and that happens more so for coding RNA molecules that produce protein, and the coding frame is basically indicated by these colored parts here. And less so, although still in majority, for long non-coding RNAs, which is another type of RNA molecule that doesn't produce protein, but that sort of has its own little function in the cell. Okay, so as Bobby mentioned, we're hiring people. So if you're at Weill, you know where we are and you know where Rosette Island is. If not, this is where we are. So this is Manhattan. And basically, we're sort of lying on three different directions in the lab that we're trying to put together. That is sort of new assay development, so new sequencing approaches, then an interest in disease of the brain, and then algorithm and software design and data analysis that comes with these new types of data that we're producing. And just before I want to go into any science, I just want to point out that there's a lot of people to thank. So that should have appeared also only on the last slide. I apologize for that. But so this is the lab. We have Anushka from India, Simon from Australia, Julien from France, Wen and Bei from China. Then Chi Lam, who's also from China, but actually currently in Taiwan. She should be with us already, but yeah, because of COVID, she can't really travel right now. And then Nathan and Eduardo are both American. Now, I'll start with mentioning some publications that we did during my postdoc times at Stanford, and I shared first authorship on a couple of papers with Fabian Gruber, Donald Sharon, and Freshta Janbani. Then I mentioned our first paper from New York City, and here are the authors of that. And then I'll go quite a bit into work that Anushka has led, but there were really many people involved in it, so you can look it up on the bioRxiv. It's a long author list, but she was really the one who pulled it all together. Okay, so quick introduction about splicing. Splicing is the process by which we take the gibberish that is pre-messenger RNA, decide what exons are, remove the introns in between, and get a final message. So here the message is, it was night in October, it's a fairly easily understood message. Now the cell may very well go back to the point where this intron was removed, and realize that there may be another exon that it could splice in, remove the introns around it, and get a second message. Hey. Okay, I'm in a call right now, okay?
French toast for lunch yesterday.
Okay.
You're very surprised.
I was just informed that there won't be French toast for lunch.
Oh, that's sad.
So here we get a second message. It was night in the... Okay. Well, here we get a second message. It was night in the lonesome October. And so this is very similar in terms of the meaning, but there's a connotation now for that message that that actually makes it a phrase out of an Edgar Allan Poe poem. So it carries the connotation of Edgar Allan Poe that the first one does not. And so that's one way to think about alternative splicing, that this sort of more or less similar messages, but slight changes in connotation. But that's only one way. So so here is an example, the FAS receptor. You have two isoforms that differ by the inclusion of one alternative exon. I'm sorry, I just closed very briefly the door.
No worries.
So there's two isoforms, and they differ by one exon, one of them which encodes a transmembrane domain. And so one of these isoforms has a transmembrane domain, the other one has not. And this isoform then will sit on the cellular surface and be a receptor. And when the apoptosis signal comes, the cell will happily accept it and commit suicide. Whereas when this other isoform is expressed, it will be a soluble protein which, quote unquote, floats around in the cell. And when the apoptosis signal arrives, the cell will simply ignore it. So it's really a question between life and death, right? So that also happens quite a bit in terms of splicing. And there's data out of Harvard where they actually showed that here they defined a dissimilarity in terms of function of protein isoforms from the same gene. And they looked at multiple isoform pairs, and they showed that for basically quite a lot of isoform pairs, there's quite distinct function. Okay, so that's something to keep in mind. I guess there's a lot of people here that are very interested in imaging. If you are really interested or not so interested in splicing and isoforms, just keep in mind that sometimes the same gene can do very distinct functions or can carry out very distinct function. Okay, so and now what I'll talk about is when we have multiple alternative exons, how are they actually combined to make full-length isoforms? And there's a couple of papers here. These are Stanford Times paper, and this is here from New York already. And I introduce the subject a little bit more. If you consider that this phrase here, I think this is a problem. And now you have a second version of it with two negations. I do not think this is a problem. Again, these boxes here are exons, and the lines in between are introns. Now, you have two negations, but you only make two versions of that gene, which are skipping both exons or including both exons. You don't make the version that is just inclusion of one and skipping of the other. What that means is that there's a statistical, or in this case, since I have phrases, it's also semantic dependence between these two. And you can look at it another way, that these two negations sort of cancel each other out, and therefore this means something very similar to that. So that is the semantic dependence. Of course, this canceling out, that doesn't transfer nicely to biology. Now, and what was not known for a long time is how these exons would be combined into full-length messages. Now, when we're in a situation where you have a strong tendency to produce only a subset of the combinations that can theoretically be produced, then we talk about coordination, okay? And so, as opposed to random pairing. Random pairing would mean that every combination at some point is made, and it's determined, the probabilities of each exon to be included would determine the final probability of all the combinations. So here you see a long-read sequencing technology that we developed for RNA-seq. Every line is one molecule. Unfortunately, I seem to have lost my cursor here. So every line is one molecule. These things here the boxes are exons, the lines are introns, and this is what we would call a constitutive exon. I should rather use this as an example because it appears in pretty much all the molecules, okay? Then here, these three I would call quasi-constitutive. There's a couple molecules that skip them, but then there are really these bona fide alternative exons, which are included in this one, maybe 50% of the molecules, and this one, maybe 60% of the molecules. What you will observe again is that a lot of molecules actually use both of these exons. Okay, so that's about 50% of the molecules. And then a lot of molecules use none of these. And that's actually much more than you would expect by random pairing. So here you have a probability of 50% of including this exon, 60% here. So by random chance, it should be 30% that include both, but you observe 50. And here it should be 50 times 40% 40% probability of skipping. So it should be 20 skipping both. You observe 30. So it's a lot more than you would expect under random pairing. So you can do statistics on that and show that this is really not what is expected by chance. And that means somehow these two exons know what the other one is doing, or at least one of them, quote unquote, knows what the other one is doing. And so in a later paper, we showed that this is actually quite frequent. So for 40% of genes that have at least two alternative exons, and here we looked at exons that are separated by constitutive exons, you have this non-random pairing, this coordination. And that is important because it means that for 40% of the exons, 40% of the genes, you can only observe their true nature, meaning the full-length isoforms that are being made, with long-read RNA sequencing. If these numbers were all at 0 here, then there would be no justification to use long-read RNA sequencing for transcriptomics. However, since these numbers go that high, tells you if you do short-read sequencing, the Illumina kind, for example, then you would be wrong in isoform quantification in about 40% of the genes. Now, I should point out that there are other papers that are quite interesting. So this came out just a couple months after ours, so it would have been done exactly at the same time. Very similar results, different exon sets they looked at, but they came to a very similar conclusion. And then there's also a paper from Argentina that was published in 2005. And it actually explains a mechanism how one alternative exon can know what another one is doing. Right. And so one thing we noticed is that when you have these coordinated exon pairs, usually both exons of that pair are entirely protein coding. We showed that later on, or different data set, that when you have this coordination, so non-random pairing, only in about 11% of the exon pairs, you have a single non-coding nucleotide. If you look at all non-coordinated exon pairs, that's about 35, I think. So it's quite a bit, quite a drastic difference. And that brings us to a model that very likely these events of coordination are there in order to produce protein molecules in which you have distant peptides, so that are very distant in the amino acid chain, where either both of the peptides are used in that protein or none. And in another protein, it could be one X or the other. So that's also a form of coordination. Now, there's a caveat that we actually don't know if all these RNA isoforms are equally translated. If they were, then we could really say that this model that I just proposed is true. For now, we have to say it's true under the assumption that all these RNA molecules are equally translated, and I hope we'll be able to check that on a gene-by-gene basis somewhere in the future. So just to give you some examples that are important for neuroscience, if you work in neuroscience, you probably heard of MAPT, maybe not MAPT under that name, but you for sure have heard about tau. Tau is basically a protein that underlies many diseases that we collectively refer to as tauopathies. And MAPT is simply the gene that encodes tau, microtubule associated protein tau. So again, every line is one molecule, yet another technique that we used for long-read transcriptome sequencing. The blue parts here are the introns, and then you see these two alternative exons here. They're both protein coding exons. And what you see is that you have a lot of molecules that skip both of these exons, a lot of molecules that include this exon and skip this exon, just a tiny bit of molecules that skip this exon and that include this exon. What you never see, or almost never see, is the inclusion of both exons. Okay, so MAPT, and that means tau splicing, is governed by coordination. And so I just emphasized to you that that coordination is often linked to the coordination of protein coding elements, exons. Now, I just want to point out that that's not always the case. So here are some other examples. Here you have a non-coding element, which is the transcription start site. So you can drive this gene from this TSS and from this TSS, the polymerase would go that way. And what you see is that when it's driven from this TSS, you have a lot of inclusion of this exon here. When it's driven from this TSS, you see pretty much only skipping. And again, that also this paper from the Netherlands that I referred to also found that. So here's a summary, long-read sequencing technologies, and there are some papers, including some papers that I was not involved in, like this one here. Then these the coordination investigation. Again, lots of papers. This one is the one I was not involved in. That's the paper from Argentina. Actually, another paper from Argentina that's also very, very interesting in the, if you're interested in coordination. And here regarding the protein coding analysis. Now, that was the time when I started my own lab at Weill Cornell as part of a neuroscience department. So we really started to think a lot more about this beast here. And then in reality, that means we often also work with the mouse brain. And given that you're a neuroscientist, it would come as a surprise to you that the brain is a highly complex mix of different cell types, neurons, oligodendrocytes, astrocytes, microglia, here also ependymal cells. I didn't put all the blood vessel cells that are also very important in the brain and its diseases. That means if you measure protein, RNA, DNA methylation, fundamentally what you're measuring is a linear combination of signals coming from these distinct cell types. And that's something we wanted to untangle. And we came up with a method that we call single-cell isoform RNA sequencing, abbreviated as SCISA-seq. The SCISA stands for precisely cutting away one cell from the others, not so much for cutting RNA molecules. That's something that we don't really do when we go for long-read sequencing. So I have this little cartoon here. This is to motivate things a bit further, and I hope you'll bear with me for maybe 2 minutes and you'll see where I'm going with that. So let's say we're at a murder site. So here's a victim lying in his or her own blood. And let's say we are the policeman or policewoman who's charged with cleaning or clearing up what actually happened. And so here's a person that gives us some information. It may not be the most useful information. I didn't do it. Here's another person that becomes a little bit more useful by pointing at someone else, accusing someone. And now this information becomes really useful because that person is actually admitting it. So we have a witness, we have a confession, case closed, another bystander. So let me put some boxes around these words so you see what I mean here, that these words are exons and the following phrases are messages or isoforms. Let me furthermore point out that there are some parts of the isoforms that are constitutive. So the did appears in every single molecule, the it appears in every single molecule, and then there are other parts that are alternative. So here the not do is alternative in that it arises in this message, but not in these two. And then the 5' end, or the transcription start site, or the first exon, whichever way you want to put it, the he, I, you also see this alternative usage. Okay, so let me furthermore give some names to these people. So we have Oliver oligodendrocyte, Alfonso astrocyte, Mickey microglia, and Newman neuron. The neuron is dead, so we're here in some topic related to neurodegenerative diseases. And so the important statement is that if we were to record only the very end of each message, we would actually lose our understanding of what happened, because now everybody's just saying it, and so we don't actually understand anything that happened. And this is sort of akin to 3' single cell sequencing. It's a very broadly used technology these days, where you go into single cells, record, you group them into clusters. So for example, into oligodendrocytes, astrocytes, and neurons. And then you look at the 3' end to check how active a gene is. On the other hand, if you were to record the entire messages, but you wouldn't record who actually pronounced them, we would equally not understand what happened here. We would know that there would be an accusation. We would know that there is a confession, but we wouldn't know who actually confessed and who accused whom. And that is analogous to long-read sequencing in bulk tissue. So we thought that we could do something really important in between. And so we came up with this technology that we call ScissorSeq. And we basically start with a single cell suspension. In our case, that was a P1 cerebellum, so 1 day after birth, and we were interested in this brain region called cerebellum. You go through the regular short read approach that other people also do, and what you can find is that then that these barcodes, and I'll come back to what barcodes are, that represent single cells. So here you have each BC1, BC2 is a single cell, and they're grouped into groups of cells that we identify with cell types. So here you see that these individual 2 cells belong to 1 cell type. Okay, so I look at this some more in this Tisney's plot that you may have seen in single cell representations. So every dot here is 1 cell. They're grouped together by similarity of gene expression activity. And then we look at marker genes. And for example, here for this group of cells, we have that they express very often TMEM119, and therefore we call them microglia because that's a well-studied gene that indicates microglia. And there you have other genes, PCP4 for Purkinje cells, very important neuron type in the brain. And so what we then did is to use an aliquot of these cDNAs for long-read sequencing. And so you have basically then a full-length long read, and at the very end you have these barcodes. And what you will do is you will, according to this barcode, you will look up in this table what individual single cell it comes from. Now this is the individual cell, which already looks like an immune cell. And this cell, we know according to this table again, comes from a cluster that we already labeled microglia, so all is good, and we know that this is a microglial isoform. We can do it then for another molecule. We'll trace that according to the barcode to this individual cell, which comes from a cluster that we labeled as Purkinje cells. So, and the first thing when you want to do that is to make sure that you're actually getting these barcodes right. And we did this first through a simulation experiment. Here we took real barcodes that we already knew to exist from the Illumina data. We simulated an error profile of the long reads. And when I say we, this was done by a student called Anushka. I will come back to her later. And by the way, this paper was led by Ishan Gupta and Paul Collier. So we introduced these errors, and then asked how often do we actually find a wrong barcode? How often do we get a wrong barcode identification? It turns out our specificity is 99.99%. And then one thing that I always tell people in the lab is when we simulate things, it's very important to realize that you simulate what you think is happening in a test tube. And that may not be exactly what is actually happening. So it's always good to also run an experiment. And so what we did here was to construct a cDNA molecule with 1 fixed barcode. So that means we know exactly what we should be finding. We put that on a sequencer. We ran the same code that we used for our barcode identification in reality and asked, do we get any false assignments? And again, specificity is 99.99%. However, the sensitivity was not as good, and for a long time I thought that that was just a game of sensitivity versus specificity. You know, it's very often when you in these sort of prediction methods, if you get very high specificity, you pay in sensitivity, or vice versa. And that is sort of what happened to us, or it looked like that was what happened to us, that the Illumina sequencing assigned barcodes to 74% of the reads, and we only got to about 56%. So that was yeah, a question why that actually happened. And now it turns out that there's chemical reasons for that, or that are in the experiment, in the single cell experiment. And I think we've solved it now. Yeah, it's not yet published, but I hope we'll come to that soon. So then a very important point was to actually show that what we get in our single cell data can be confirmed or validated with other data. And we were especially interested in validating it with data that wouldn't have any PCR, because PCR can actually cause problems in your results. So what we looked at is mass spec data, so proteomics data, and that was in collaboration with Frank Koopmans and Guus Smit. And actually we did further validations with Stephen Sloan and Ben Barres, and I don't think I have their slide for today. Unfortunately, Ben Barres passed away before he could see the outcome of this work. Anyway, so if we consider junctions in our single cell data that we only observed in microglia, so these are microglia specific junctions, and we look at mass spec data from proteins that come from neurons or from microglia, we see a lot of signal that's spanning these junctions in the microglia, really showing that our microglia specific junctions are well called. And then when we look at neuronal specific junctions, we of course see the opposite. Now, beyond just validating this approach, it also means that we have some predictive capacity for what's happening to proteins in terms of isoforms in these single cells that we look at. And that is very important because fundamentally for protein coding genes, at least, we're usually assuming that what we observe on RNA level means something on protein level. So it was very important to actually confirm that we can make these this extrapolation, at least broadly speaking. So what you see then here is that we can take all these single reads. So again, one line is always one molecule. We can group them by types. So here I have one type of neurons in blue, another type of neurons here, and a third type of neurons. And then we have astrocytes, microglia, and OPCs. In orange, the alternative exons are highlighted. And what you can see is that that, for example, this exon is included in all the neuron types, so is this and this. And what happens then is that the neurons are basically characterized by two isoforms, the one that include 1, 2, 3, 4, 5, 6 alternative exons, and the one that just skips the 5th. Okay, you see that?
Hagen, I have a quick question, just for my own clarity. When it comes to these exons, are all the ones expressed identical in one of these genes? Or are they still vary between each other, even though they're exons?
So you mean, does every exon have the same RNA sequence?
Right, yeah.
No, they're completely different. So
I see.
I mean, there are some that share some similarity, but let's say if you know the sequence of this exon, it virtually has no predictive value for the sequence of that exon.
But yet they all show up around the same length, like position on the gene.
Yes and no. So what I did here is the length of these exons is drawn to scale. So I would say that it is somewhat an uncommon gene in the sense that the variation in the length of these exons is relatively small. Now this one is really small here, but overall, if you go genome wide, there can be huge variation in terms of the length of exons. So it's somewhat misleading here that in this example, the exons are mostly of the same length. Yeah, I never actually thought about it. Now, just one thing I want to point out, there's even more variation in the length of introns. And here I actually drew all the introns as if they had the same length. So the introns, I faked them into having the same length. And so this one is longer because there's an exon in between. So it's actually these two things here that have the same length. Okay, so in these two isoforms that characterize the neurons, this one here, the inclusion of these 6 exons and the one that's skipping that, you don't really see that in astrocytes, microglia, and OPCs. So what that means is that there is cell type specificity in the isoforms that is not there at gene expression level. Okay, and you know what? I think I'm going to skip over this here, just because in the interest of time. So what we made then is this this database that we call isoformatlas.com, where basically you have all these isoforms listed. Sometimes they have an identifier which we call which is called ens-must-t, which basically means coming from Ensemble. That's a very or GenCode in this sense, in this case, which are very famous databases for isoforms. Must, because we're working on the mouse, and T for transcript, and then there's a number. Now, sometimes you will see that there is different IDs, and that is the case when these isoforms were actually not present in the databases. And here you see one of these examples. So it's a novel isoform, and it's the major isoform in astrocytes, OPCs, and microglia. Then you see another isoform, again, the novel isoform. It's the major isoform in Purkinje cells, and two other types of neurons. So here are the Purkinje cells and two more types of neurons. And here you have the astrocytes and OPCs and microglia, and you don't really see it expressed there. Okay, now the following is in progress, so please don't scoop us. It's on the bioRxiv, and it was led by a grad student, Anushka Joglekar, and all of that is coded in an R package, which we call ScissorSeeker. And basically the question that we followed up on here is, when you have differences in isoform abundance between hippocampus and PFC, or any two brain regions, or any case control studies, what does it actually mean at a single cell basis? Now here we chose hippocampus and PFC during early development. It's a time when there's a lot of growth happening in the brain, and so we sequenced this quite deeply. So there were something like 15 million reads that, I think 13 probably, that were longer than 500 bases, about 7 million that are longer than 1,000 bases, 5 million that are longer than 1,500 bases, and obviously it gets less as the reads get longer. And the first thing that Anushka did was to think about the statistical framework of determining differences in isoform abundance between two cell types. And so if you have a hypothetical gene here, and there's two alternative exons, and let's say there's these 4 molecules being produced. If you think back of the coordination and the random pairing I talked to you earlier about, here that's clearly random pairing, because all all combinations are made at equal levels. Now, what you can then realize, is that you have for the red exon, you have 2 reads supporting its inclusion, and you have 2 reads supporting its skipping. Okay, so that means you can say there's 2 out of 4 reads use it, so 50% of the molecules use it, and we refer to that as PSI or psi percent spliced in. And now in a statistical framework, what you can do is you can open up a 2x2 contingency table, for example, looking at 2 conditions, for example, 2 cell types, or healthy and diseased, or 2 brain regions. And you can record how many reads you have that support the exon being included and that support it being skipped. You get this 2x2 contingency table, and you can now run a Fisher test or a chi-square test, whichever one you like. The statement is now, since you have 2 exons in the same gene, you would get 2 tables, and that means the tests would be dependent. That means you have to correct for multiple testing using the Bonferroni or the Benjamini-Yekutieli correction, and those are pretty harsh. Now, what we can do now, since we sequence full-length isoforms, we can reason in a world of isoforms. Okay, we can say that for this isoform, we have 1 read supporting it, and then we have 3 reads supporting another isoform. So it's 1 out of 4. That gives us a percent isoform usage of 0.25 or 25%. And we abbreviated that now as pi. So percent isoform as pi gives us another Greek letter. Now that means we will now get a 2x(n) table where you have, again, the 2 conditions, and you will have counts for each isoform. There can be many of them. The important point is now that you get exactly 1 test per gene. Okay, and so that makes these tests independent. They can also take into account not only the exons, but the very beginning and the very end, so the TSS and the poly-A site. And now you can correct for multiple testing using the Benjamin-Hochberg correction, which is not as harsh in terms of numbers. So once you have this concept of pi, you can define a delta pi between two conditions for the for each isoform, and now you can aggregate these isoform delta pi's to the gene level. Now you want to be careful because if you add up all the delta pi's, they will simply add up to 0. Because whenever one isoform goes up in a condition, another one goes down. So what we do is, that means we want to add up the delta pi values of isoforms that we think matter. We also don't want to add up too many because there are genes where there's thousands of isoforms, not that often, but that exists. And so you don't want to add up 100 very small value to come up with one large value. So what we say is, well, we want to have a delta pi on the gene level that is contributed by at most 2 isoforms. And so once you have that, you can plot this volcano plot, which is quite common in genomics. So you have the delta pi value for each gene. So each dot here is a gene. And then the y-axis tells you how significant it is. And so in red, you get the things that are significant, the level of significance here. And on the x-axis, you get how strong the change is. A value of 1 would mean a night and day shift from one isoform to the other. And it turns out that when Anushka did this, when she did the tests on exon level, she found 30 significant, so 30 genes with significant exons. When we do this on isosome level, we find almost 400. So that's really an improvement, because now with a set of 400, you can actually do things afterwards. Now, in I think around 80 cases, there were novel isoforms that were part of this change in regulation between the two brain regions. And we collaborated with the GenCode people, and they validated them manually, and they validated every single one of them. So that means for those isoforms of high biological interest, we actually now have an automated way of validating that. Okay, so now the second question is then, if we have such a difference between two brain regions, what cell types are actually underlying that? And it could be one cell type, like for example, excitatory neurons. It could be multiple cell types, or it could be no cell type at all. And that is because a brain region comparison, or comparison of two samples, often is actually an apples to oranges comparison. In the sense that you might have one sample which contains a lot of cell type 1 and not so much of cell type 2, and then the other sample may do the opposite. So that means you may see differences in the bulk, which is sort of the linear combination of the cell type signals, that don't really exist in any of the cell types, just because your bulk may be dominated by different cell types in one condition compared to the other one. So here I just want to go through examples. I think that's more more teaching actually. So here again, every line is one molecule, and here we have the data coming from hippocampal excitatory neurons. And you see that this exon is pretty highly included, maybe in 80% or 90% of the cases. When you go to astrocytes, it's still pretty high, let's say 50%, still in the hippocampus. And then when you go to OPCs, here it's also pretty high, maybe, I don't know, 75%. You go to the same 3 cell types in the PFC, and you don't actually see this exon included. Okay, so here it really seems that that the brain region is overriding cell type specificity. Now, and what we observe is that this multiple cell type model, or where brain region overrides cell type specificity, is observed more often than you would expect by chance. But it's very important to point out that this does not at all represent the majority of cases. Okay, so it's a small number of cases, but clearly more than you would expect by chance. So that means we think for those cases, it actually matters. Now, here's another example. So we look at hippocampal astrocytes and PFC astrocytes, pretty high inclusion of these 2 exons. We go to OPCs in the 2 brain regions, again the same. And then we go to excitatory neurons, and we have about 81% inclusion in the hippocampus and 23% of inclusion in the PFC. So that means there's a quite drastic change. It's clearly dominated by one cell type, which is excitatory neurons. And obviously this can be broken up more into finer subtypes of excitatory neurons, but this then becomes statistically more challenging because every time we break up a cell type, we lose reads. So that does, this example represents the majority of cases. Okay, so as a summary, in the majority of cases, there's a single cell type that actually explains brain region specific splicing. Now, but this multiple cell type model, we do observe more than by chance, and there's no cell type at all, at least in the comparison of these two brain regions, is rather rare. So now I'll talk a little bit about isoform diversity between cell types in the hippocampus. And so that matters in brain region comparisons because there are some cell types in the hippocampus, or at least in the hippocampus sample that we have, that have no equivalent in the PFC. So the hippocampus is relatively closely located to a region called the choroid plexus. And so when we do a hippocampal isolation, we actually get quite a few cell types from the choroid plexus that we don't get in the PFC. Likewise, there's a lot of for example, radioglial cells in the hippocampus that at least we did not observe in the PFC. So what you see here are pairwise comparisons of 4 high-level cell types: vascular cells, myeloid cells, neuroglia, and neurons. And this percentage gives you the percentage of genes that changes isoform abundance between these two cell types. And you see that, for example, between neurons and vascular cells, it's huge. Okay, and then myeloid cells and vascular cells, not that much. And here it's broken up by further subtypes. So the neurons are broken up by inhibitory neurons, excitatory neurons, and granuloneuroblasts. And then the glia are broken up into choroid plexus cells, astrocytes, RGLs, NIPCs, and oligodendrocytes, and so on. And more or less this confirms that. Now, when you make comparisons of 2 neuronal cell types, for example, 2 types of excitatory neurons, then you usually observe something like 9% of the genes having differences in isoform abundance. At least that's where the peak here is. When you go into when you compare 2 non-neuronal cell types, then the peak is here, I don't know, 16, 17% maybe. So that means there's more isoform diversity among non-neurons than there is among neurons. Some people may see this as surprising, others may not. Nevertheless, it is the case. But what's really surprising to us is that this blue distribution, if you split it up by whether one of the 2 cell types is choroid plexus epithelial cells, which you abbreviate as CPE, there's a huge difference. So when these CPEs are involved, there's huge differences in alternative splicing with maybe 23, 24% of genes usually showing differences in isoform abundance between the CPE cell type and another glial cell type. When it's to other glial cell types, so that none of them are CPEs, then it's much less. So really these CPEs seem to be very, very different. They seem to be the outlier. And here we can then trace back where this difference in isoforms came from. We see quite a bit happening at the exons, so internal exons, quite a bit at the poly A site, but the largest differences were really observed at the TSS. And yeah, this is the summary, and really pointing to the fact that these choroid plexus cells are quite different. Actually, what I didn't really mention, but the strongest isoform difference is actually what you observe when you can compare a neuronal to a non-neuronal cell type, which may not be that surprising. Okay, so now I'm going to talk just a little tiny bit about functional consequences. So this is a gene called FGF13, and it's a gene that we are highlighting here because we get very fortunate to collaborate with Susan Lin in the PIT lab, so it turned out that FGF13 is the gene that they're working on, and it was more or less highly expressed in our data, so we could actually make some statements about it. And here we see that there are 2 isoforms, the S and the VY isoform. The color code here means what the relative abundance is in the different cell types. So the yellowish color means almost exclusive usage. And I'm losing my cursor here. That you observe here in these excitatory neuron types. And then in the inhibitory neurons, you see more of a mixture, the exception being Cajal-Retzius cells. And so what Susan then did is to look at the subcellular localization of these isoforms. And here what you can observe is that the S isoform is really located in the nucleus, whereas the VY isoform is located all through the cell, including the prostheses. So that really points to important functional differences, because obviously the protein can't exactly do the same thing in the processes that it would do in the nucleus. And we're very grateful that this could be added to the paper, because it could really allow us to see the implications of these isoform changes. And so we have this database. In the old version, we had 10 cell types and not that many counts. Now we have 10-fold more data and 45 cell types across 2 brain regions. And these cell types are grouped by inhibitory neurons, excitatory neurons. So there's multiple types of excitatory neurons. I didn't really go into that in the talk, but you can really go into the precise subtypes: granuloneuroblasts, glia, myeloid cells, vasculature. And there's so much data that it may be a little more difficult to look at. So here I'm going to blow this up, and what you see is the gene CAMK2B. What you see is that this isoform here represents about, I think, 80% of the molecules in this one type of excitatory neuron. So we call them type 2 in the PFC. And then you go into another type of excitatory neurons in the PFC, and there it represents about 40% of the molecules. And then you see other isoforms popping up that are very rarely expressed in the other type. And then if you go into hippocampal interneurons, then it's a completely different picture again, where this isoform becomes the dominating isoform. Okay, so it's if you're interested in isoforms in the brain, this is quite good resolution, and we're going to be adding more data from other brain regions and developmental time points. So the last point that I want to talk about is spatial isoform sequencing. This is based on a technology that is marketed by 10x Genomics, called the Visium technology. And basically what it does, it uses these barcodes that I mentioned earlier, not to identify the RNA molecules of single cells, but to identify the RNA molecules in a certain spatial location. Okay, so here every dot is basically giving one barcode. That means you get something like a couple thousand, in this technology actually more than 10,000 molecules from each spot. Okay, and that means afterwards you can check whether a gene was expressed rather in cortical areas, or in the hippocampus, or in specific layers, and so on. And so what we did is we developed a long-read sequencing approach for that in order to measure splicing and isoform abundance. And so what you may recall from the earlier part of the talk is that we had single cell isoform expression from hippocampus and PFC. So that means we can, for each exon, we can calculate a delta of its usage between hippocampus and PFC. Okay, so that's what we here did on this axis. Now, we didn't have a slice of brain that actually included the hippocampus and the PFC. But what we have here is a slice that includes the hippocampus and then many parts of the cortex. So we used here the cortex as a proxy for the PFC. And that means now, based on this cortex and this hippocampal data here, we can calculate a spatial delta. And what it actually shows is that these correspond quite well. Not perfectly, but quite well. And that means we think with a spatial technology, you can actually do quite interesting things. And so what you see here is a couple of genes. So CLTA, which I believe is clathrin A. I should double check that. This exon here, yellow again means high inclusion, and you see quite high inclusion in the hippocampus, and you see quite a lot of skipping in the choroid plexus. And then this other exon is actually skipped pretty much everywhere, but there is some inclusion in some specific parts of the hippocampus and some very tiny pockets here. And then another gene here, PKM, again, high inclusion in the hippocampus as seen by these yellow dots here overall, and then skipping in the choroid plexus. And the other exon here, really does pretty much the opposite. Okay, and then another example here, SNAP25, which is characterized by two isoforms, the A and the B isoform, and pretty high inclusion in this area, and then goes towards lower inclusion in the hippocampus and cortex and cortex plexus areas. So there's really this gradient, but there are pockets that really defy this kind of gradient view. So spatial isoform sequencing allows you this really to pinpoint isoform abundance to spatial locations, and there's some global trends and some highly precise exceptions to these global trends. Right, with that, I'm done. I want to thank you for listening. Now, I did my PhD in Barcelona, and I'm very thankful this really got me started there. This was in Roderick Giegold's lab. Then postdoc days in Stanford, and I shared first authorship with these three authors. Also worked a lot with Morten Rasmussen, and this was done mostly in Michael Snyder's lab. Ben Barres helped me a lot, although formerly he wasn't my advisor, so I'm very thankful for that, because he actually had no obligation to do so. People at PacBio, at Illumina, that have helped with the data sets over the years. People at Weill Cornell that helped me quite a bit. Collaborators in Amsterdam and Emory, the authors of the Nature Biotech paper that I mentioned in the beginning. Then the paper under review now is it's really a lot of authors, but it was led by Anushka Jogalkar. This is the lab, the funding sources, and yeah, if you have questions, then please ask away.
Thank you, Hagen. So I'm really glad you touched up on the spatial mapping, because that's all I was thinking about. Because I know people had efforts where they want to map gene expression to cell types in the brain and look at the spatial distribution. So I guess what you did is the step before that, which is to map isoform expression throughout spatial regions.
Exactly.
Has there been efforts in using like these isoform data to predict spatial distribution of cell types?
Not from the isoforms. There have been other people that have done that from gene expression.
Yeah, exactly.
And it works actually relatively nicely in the sense that you can get some sort of predicted composition of each physical spot. Now, I would say right now, there's still, each spot here is still quite a few cells. So in a sense, you always expect that there is at least some astrocytes and there's some oligodendrocytes and there's some neurons. However, of course, if you're thinking about localizing very specific neuronal subtypes, I mean, that could, I think, very well be done. Now, from the isoforms, we haven't really done that yet, just because the data is not that deep. Right? What we can do is we can really show you that some isoforms are more representative of the hippocampus as opposed to cortical areas or the cortex plexus or also other areas. But the isoform data is always a little bit less deep than gene expression data. So yeah, we haven't predicted cell type abundance from the isoform data.
Yeah, I hope the database for isoforms keep growing, because there's so much. Yeah, if it gets deep enough, we could do so much modeling and so much predictions. Karine had a question. Do you want to just unmute yourself? And
yeah, that was a great talk. Thanks. So I had a question about the scissor-seq and some of the other analyses that you talked about near the beginning. And so when you're doing these, are they only restricted to mRNA, or if not, how do you, or do you differentiate between protein coding RNA and other kinds of RNA and their isoforms?
Okay, so I was actually, for a second, I was misled by your question. Now, when so everything relies for the moment on poly-DT primers, okay? What these poly-DT primers do, they will pick up in principle every molecule that has a poly-A tail. Now, that includes a lot of long non-coding RNAs. There is, it's a statement in the field that is that I think is often sort of maybe misunderstood a little bit, that long non-coding RNAs often are not polyadenylated. there is undoubted, well, without a doubt, there is long non-coding RNAs that do not use a poly-A tail but have a different structure at the 3' end. However, a lot of long non-coding RNAs also have poly-A tails, so they are also getting turned into cDNA, and therefore we're investigating them as well. However, long non-coding RNAs are very lowly abundant. So if you do it the genome-wide way and basically turn all the molecules into cDNA, then 98% of your molecules will come from protein coding genes. So that means so the long non-coding RNAs will not be the majority of molecules. Now there's also a lot of efforts in the field to investigate other kinds of molecules. And we and others are working on these things. So there is, of course, interest in that. Yeah.
Out of curiosity, has there been, like, because I'm not really familiar as to why some isoforms are more frequently seen than others. Like has there been any links between epigenetic changes to these patterns, like alternative splicing patterns? Is that a cause? Like
the genetic basis. So when you mean genetic, you would mean mutations or something like that that would influence isosome abundance, or do you mean what are sort of the how does a cell actually determine which isoform to express?
I'm just curious because like epigenetics has always been like super interesting, and it's not really like a mutation, right? It's more like a silencing or has that been like implicated in causing some of these isoforms to be more expressed than others?
Yeah, of course. So there's a whole field of splicing regulation. And I mean, there's a lot of what we call splicing factors that, so these are protein molecules that bind to the RNA and that determine where exons and introns are. And there are multiple levels. There's sort of what we call the core splicing factors and the auxiliary splicing factors. And it's multiple hundreds that the genes and proteins whose role it is to splice RNAs in the correct way. And so, for example, different expression of splicing factors in different cell types is linked to different isoform expression in these cell types. Okay, so that's one very clear level. Now, if you, since you mentioned genetics, there's also quite a few examples where we know that, for example, mutations in cancer can cause difference in isoform abundance or difference in isoform expression. And that can be either in the target gene, so the gene itself has a mutation that causes some difference in splicing. But one thing that's also known is that it affects that there are a lot of mutations in splicing factors, so that that means they then start to missplice genes, and basically the mutations in a reduced set of splicing factors then affect many downstream genes. And I guess if you're not in the splicing field, you can think of the whole process a little bit like transcription, right? There's transcription, there's a lot of transcription factors that influence initiation of transcription and elongation. And to some extent, you can think of it exactly that way. It's of course a very different process and things go differently, but in a nutshell, cell type specificity is, at least to some extent, defined by these cell type specific expression patterns of splicing factors. And what's actually interesting is that the splicing factors themselves are heavily alternatively spliced.
That's, yeah. I can only imagine how many things people can do with more data. And I think BrainHack is such a good opportunity to spread this sort of knowledge.
Yeah.
So
just to point out that there's a real need to get so there's an experimental part, but there's also very important analytical and algorithmic parts to this work. So
yeah, I mean, I'm hoping one of the future events, one of your grad students can submit some sample data and a project and drag some more brain hackers into it, because that would be amazing.
Okay, we will do that.
Okay, if there's nothing else, I'm gonna stop recording and I'll push this out to YouTube after the event.
Okay.
Yeah, thank you for coming, Hagen.
Yeah, thank you. And